Good morning, everyone. How are you doing? I hope well. Good morning, everyone. Hope you're all doing well. Missed interacting with you in lecture. Here we go. You ready? Let's finish signal transduction once and for all. Before we do, you want to hear a joke about potassium? Okay. I was also going to tell a joke about sodium, but nah. I may look like I'm not doing anything, but at a cellular level, I'm quite busy. And you can now talk about how that's all regulated. Wow. Why did bark or beta-adrenergic, oops, I screwed up the joke. What did bark beta-adrenergic receptor kinase say to Clathrin about beta-adrenergic receptor? This one we'll actually talk about today and is in the textbook. Arrestin. Like arrest him. I made that one myself. I could probably tighten up that joke a little bit. Yeah. So close, so close. By the way, I'm not a boomer. I'm actually a millennial, but that's okay. Okay, boomer, here we go. I've posted the in-class activity for this week on Moodle. It's due this Sunday by midnight. Again, I'm just trying to encourage you to interact with the material so that come the final, even though it's take home, you will be able to complete it within a time frame that I've yet to determine. So please keep up. I understand. I do understand. Like, this is not the priority in a lot of cases, and I totally get that. But I totally get that. But at the same time, I'm trying to give you a solid finish so that you can say like that you really had biochemistry, a solid semester of it. So I'm doing my best to make this interesting for you. I noticed some people have already started posting discussions about progeria on Moodle. Some nice back and forth going on there. Keep that up. It's fun to read. I encourage you to also try and answer the last two questions, which deal with actual biochemistry. Use it as a forum to ask questions as a class, and I'll interject if I see you're at a roadblock. But these two questions deal with the journal club paper, the Zhao Quinn et al., where it's asking you to provide an explanation for why the drug lonafarnib isn't effective, or talking about the nature of the most common mutation in progeria that involves cryptic splice sites. These are areas that are like moving into the realm of truly real life, like real life applications, but it's inherently complicated. So I can't force you to, but if you'd like, you can utilize the online forum to like be like, hey guys, I'm confused about this. What do you think? So we're, like I said, we're at the end, and I'm going to start talking a little faster now. We're at the end of the road in terms of signal transduction. We talked about the main themes underlying all of these different signaling pathways, but the one that we didn't really get to yet was desensitization or adaptation. And this is in the later parts of chapter 12, section 2, and a little bit of 12.3 as well. So I'm going to go through that briefly with you today. Hopefully I'm going to try and get today's lecture down closer to like 35 or 40 minutes. So hold on. So desensitization or adaptation. The idea that the sensitivity of the receptor itself is subject to modification. So there's a signal, and the signal is present. Stop. So what I'm trying to say is that the signal is present, but you have a different output. So in this case, if the receptor were Spock, he's getting something that's making him sad and activates the pathway. But there are active mechanisms that even in the presence of a signal, you wouldn't have a response. And that would be Spock encountering something sad, but it's just not affecting him. The signal, he doesn't turn on the signal cascade. So what if you were a cell, wanted to be less responsive to a signal? In some regards, this is review, but just keep hitting it. This will be the last time we hit it. This is the figure from your textbook. We talked about sensitivity, where the signal geometrically increases. This is a pictorial form of that, where you have catalysts activating other catalysts, where you start with one activated receptor, 10 activated, and they're just estimating here, G protein alpha subunits, which make 200 molecules of cyclic AMP, so on and so forth. So that's sensitivity. But how do you desensitize this pathway? That's where we're heading. The more general question you should be able to answer is, when does response to some signal end for G protein coupled receptors? And let's focus on beta-adrenergic receptors. So when does response to beta-adrenergic stimulation end? I don't know if there are actually 6. I think there may be 4 or 5 answers, but the first answer is, in order to end, you have to start at the beginning. So the first step in this pathway was binding of an agonist to your receptor. And if you remember back to Unit 2, Exam 2, this is where a term called KD, the dissociation constant, comes into play again. So when the concentration of agonist is lower than the dissociation constant in blood, that's when you would start to have no more stimulation of the pathway. Why? Because the agonist would dissociate from your receptor. I might review the plotting on the y-axis your saturation Y, and on your x-axis your concentration of substrate. And yeah, so this is all, this is mostly happening, like this would be, the outside is perhaps where the blood is, and it's accessing hormones that have been excreted by nearby cells or cells elsewhere in your body. But whenever the concentration of your agonist goes below the KD, the signal will start to stop. This leads to a change in what? Well, as soon as you have the agonist no longer bound to the receptor, what's going to stop happening? Well, the GS alpha, the G protein alpha subunit, is going to stay associated with the beta gamma subunits. Remember, it's the conformational change that starts here in the receptor that ultimately releases the alpha subunit. And if it's not released, it can't bind with the effector and do all this other magical stuff here. Second way, any other ways looking at this pathway that you might think this is how stimulation would end? I'm showing you a hint here. I talked about this notion of a molecular timer. It's intrinsic in the enzyme's activity to turn off. Where was that in this pathway? That was your G protein. It has GTPase activity, specifically the alpha subunit that binds guanine nucleotides. We also talked about the regulation of G proteins by GAPs, or G protein activating proteins. So both of these are getting at the timing aspect of this step, right? GTP hydrolysis makes you go back here. And if you have a factor such as a GAP that promotes that hydrolysis, you'll end up over here. Any others? I'd say shoot the messenger in this case. In fact, shoot the secondary messenger. Didn't talk too much about it, but it's in your textbook. The secondary messenger in this case is cyclic AMP, and there's an actual enzyme called cyclic nucleotide phosphodiesterase that breaks a covalent bond, and cyclic AMP can no longer bind to things to activate them. Things like, remember the regulatory subunit of pKa? So shoot the messenger. So this is biochemistry, so maybe I should show you a chemical structure every now and then. This is, this is ATP, and we're shooting the messenger. So you have your 3-phosphates, your adenine sugar molecule, and a nitrogenous base here. So adenylyl cyclase, remember the effector protein, it's kind of neat to look at the chemical structure of what's happening. It's removing the 2 phosphate groups, and then in the remaining phosphate group, it's making a covalent bond between the oxygen and the 5'. We didn't cover it in this semester, unfortunately, but you can number these carbons 1, 2, 3, 4, 5. And usually the 3' and 5' end are important in synthesizing DNA or RNA. But here we have, within a single molecule, both the 3' and 5' carbons of your sugar. In this case, it is a ribose sugar, I believe. Anyways, you can see that's why it's cyclic. So this is your messenger, and you're going to shoot your messenger kindly with Nerf bullets. The thing that's terminating it is this enzyme, and all it does is simply cleave this bond between the phosphate and oxygen at the 3' carbon of your sugar molecule. Okay, so that's shooting the secondary messenger. Essentially all steps you can go backwards. You can see the secondary messenger itself, remember, was binding to which part of pKa? You had your R, your regulatory, and your catalytic subunits of pKa. This would be, I believe, the regulatory subunit was binding cyclic AMP, and then no longer binding to the catalytic subunit of pKa, allowing pKa to phosphorylate downstream targets. So this phosphorylation event, though, it's phosphorylating things like phosphorylase B, which was what was playing a big role in secreting glycogen from your cells or tissue. This phosphorylation by pKa is reversible. So it's not shown in this figure in your book, but so this is a kinase, remember, that's the name of an enzyme that's adding a phosphate group to something like phosphorylase B, which I just talked about. But you can also go the other way. And the name of this class of enzymes is called a phosphatase when you remove a phosphate. So there are lots of phosphatases in your cells. The textbook mentions one in particular, PP1. Sorry, which stands for something. It's in your textbook. Anyways, this is another mechanism to stop the signal. So in this case, removing a phosphate turned off the pathway, but you can also add phosphate groups to other things to dampen the pathway. So the last example we'll talk about that has to do with desensitization is a kinase that phosphorylates part of the GPCR, the beta-adrenergic receptor. And so, apologies, my slides are a little messed up. All of these answers that we've talked about until this last one, the phosphorylation of the beta-adrenergic receptor, these are all ways that the signal can be shut down, but primarily when the signal itself is removed, when epinephrine is no longer present. These are the mechanisms by which the pathway will shut down. But there's a way in which you can actively shut down GPCR signaling as well as other signaling, even if the signal is present. So even if epinephrine is present in the blood at a concentration higher than the dissociation constant with beta-adrenergic receptor, for example, or in last week's example, even in the presence of a, well, we won't go there. Let's just focus on GPCRs. There are ways you can shut down the signal in the presence. There are ways you can shut down the signal transduction pathway in the presence of a signal. What is the most effective way of being insensitive to signal? Let's say someone's being rude to you. Maybe one of your siblings, or I don't know if you're at home, your mom or your dad is just up in your grill. What's the best way to be insensitive to that signal? You can't get rid of the signal. You guys are stuck in your house. Remove yourself from the situation, like physically. Get away. Go into a room and lock the door. Physically sequester yourself, right? That's what we're talking about in terms of receptor downregulation. Receptor downregulation, as shown in this slide, involves physically sequestering you, the receptor, from the situation, which is being monitored by binding of a agonist. So you can see these waves are just indicating all the magical little arrows, pathways that we've talked about. But an important way to modulate strength of a signal pathway being activated to a signal is by modulating how many receptors are at the plasma membrane. Here we just see one, but in reality, there can be many or not so many. So the first step of this is in recruiting protein machines that help bud the membrane and bring it into the cell, such as clathrin, we'll talk about in a bit. So you can have endocytosis, and then your receptor is now in an endosome, a small vesicle. We won't go through it in great detail in this class, but just be aware that it's actually not shown, well, it is shown here. Signaling can continue even inside of the cell. It's pretty well documented. These vesicles go through a very complex sorting process within the cell through the endosomal pathway. And the endosomes themselves can make vesicles. So you can have a vesicle within a vesicle. These are called multivesicular bodies. And the point of being sequestered in the intraluminal vesicle of an endosome is that things that are inside of these structures, when they fuse with the lysosome is a degradatory. It has acidic hydrolysis that degrade things inside of these structures. So we're talking about desensitization of signal transduction pathways, and it's kind of interesting because this is where biochemistry and cell biology really start to converge, where you have biochemistry of enzymes regulating one another, but there's an additional layer of sorting within compartments of a cell. Just a side note, part of the reason I went into that detail is because this arrow here of involving the creation of a vesicle that might sort a receptor inside of an endosome, that's what I did my PhD on at the Mayo Clinic. So I spent 6 years here. I studied the proteins, the enzymes that help mediate this step. But you are here, so let's come back to here for just a little bit. By here, I mean the plasma membrane. So you've already, to some extent, seen or heard of receptor downregulation, maybe some more than others if you took cell biology. But now I want to add in an additional layer of biochemistry to be rigorous biochemically. And for you to not only appreciate that receptors are downregulated, but how this is tied to a signal biochemically. So this is figure 12.8 in your textbook. We're talking about desensitization and inactivating phosphorylation of the G protein coupled receptor itself. You may have been wondering what that dog was on the previous slide. I'm trying to help make this slide a little more memorable for you. But what this is showing is that this process of desensitization that is going to involve endocytosis, you can see a vesicle is being formed here. And now the receptor is removed from the situation, right? It can't interact with ligands that are on the outside. This is mediated by two proteins. BARC and beta-arrestin. So the first step we've already talked about in great detail, where you have binding of your agonist, epinephrine, to your beta-adrenergic receptor that does what? Causes a conformational change that's propagated to the GS alpha subunit, right? That goes on to activate the effector protein, adenylyl cyclase. But you may have wondered to yourself, what about beta and gamma? Like, do they actually do anything? Or do they just like sit there and wait for the alpha subunit to hydrolyze its GTP into GDP and then bind back to the GS alpha subunit? The answer is yes, they do something. They play a direct role in desensitization. So again, we've spent a lot of time talking about the alpha subunit in terms of it having intrinsic GTPase activity. Now we're talking about beta and gamma. What do beta and gamma do? Well, they recruit this thing called beta-adrenergic receptor kinase to the membrane. And what does this kinase do? Kinases add phosphate groups. Think back to Sark kinase, Tina and Jennifer's presentation. BARC is phosphorylating the G protein coupled receptor itself. It's a serine kinase, so it's you can see this kinase here has added 2 phosphate groups onto the carboxyl terminus, which is on the cytoplasmic side of the receptor. Okay, that's all great. What does that do? Well, once this part of the beta-adrenergic receptor is phosphorylated, it now binds to something called beta-arrestin. Beta-arrestin is really important because it can interact, it can recruit other proteins called clathrin. And clathrin forms this cage-like network. It starts making this cage that eventually causes the cell membrane to bud into the cytoplasm. And then after it cuts off, after there's a scission event, you have an endosome, an endocytic vesicle. So the consequence of beta-arrestin being recruited is that you have endocytosis, and after endocytosis occurs, the beta-arrestin can at some frequency fall off, and then this will be, these phosphate groups can also be dephosphorylated by a kinase or a phosphatase. The point being that this can be recycled. It's a reversible process in terms of the receptor being present at the plasma membrane or not. So this was my joke. What did BARC say to, oh man, what did BARC say to, what did BARC say to the, I don't even know it. Oh, okay, no, this was the joke. What did BARC say to clathrin about beta-adrenergic receptor? And the answer is arrestin or arrestin. And it's kind of like being arrested and being carried off to jail because the receptor is being removed from the situation. So these are BARC and these are arrestin coming to get you. There's a lot of cool videos about clathrin. If you are looking in your slides and you want to learn a little bit more about it, click on this hyperlink here. It shows you how the cage-like network forms, leading to that process of making a vesicle. If we actually look at the structure of G protein coupled receptors, the point here is that arrestin was not only recruiting clathrin to cause endocytosis, but it also actively prevents the binding of the G protein coupled receptor to a trimeric G protein. So it cannot activate signaling by interacting with its coupled G protein. This is the beta-adrenergic receptor that we've spent a lot of time on in red. And there's a crystal structure where this is bound to the trimeric G protein alpha, beta, gamma subunits. But they have also solved the structure for a G protein coupled receptor bound to the arrestin molecule. They don't have this crystal structure solved for beta-adrenergic receptor bound to arrestin, but instead another G protein coupled receptor called rhodopsin that we will talk about a little bit. It's involved in sight. So this is the last sort of part. We're not going to go into as much detail for chapter 12, section 3, just because of time, but it is essentially what I hope you'll see is that if you wanted to, you're rapidly able to dissect any signaling cascade pathway. And I'll show you, we'll show you here, I guess, in how rhodopsin is working. The cool link here that from a biological perspective is that G protein coupled receptors, right, because they do everything, almost everything, they play a really important role. They're in the plasma membrane of sensory cells. And the vertebrate eye, at least, uses a G protein coupled receptor in sight. This is one of my favorite books. When I took evolution in college, I don't know if you've ever heard or seen it, it's by Carl Zimmer. But you can see it's showing you the different eyes and all these different cool organisms. But now, hopefully, after you take this class, you appreciate that at the biochemical level, certain cells in their eyes had G protein coupled receptors called rhodopsin. And yeah, the substrate in this case wasn't epinephrine for rhodopsin, but actually a photon, which I just can't get over how interesting that is. That photon could be a substrate. So take a break. This is taken straight from your textbook, but read this. As your eyes move across this sentence, the retinal images of the first words disappear rapidly before you see the next series of words. In that short interval of time, a great deal of biochemistry has taken place. This was a really cool paragraph in your textbook, I thought. Do you understand what this is saying? Like, you have a G protein coupled receptor initiate a signal cascade, and this is allowing you to see something. But all of these mechanisms of turning the pathway off, preventing runaway signaling, right? It gives you a feel for the time scale at which this is happening. How quickly does it need to happen? Like, insanely quickly, because otherwise we would sort of have this effect where every time you moved and looked at something else, there would be a blurred image of what you saw before, right? You need it to be extremely fine-tuned temporally. So you don't have to know all of the details of this figure, but now you should be able to understand what's happening. If I were to ask you a question, rapidly be able to pick out the relevant components of the pathway, right? So what do you think this is? This is your G protein coupled receptor, right? And you can see that its alpha subunit has a different name. It's called T. Their G proteins are called T. T alpha, T beta, T gamma. What caused you in the previous slide for these first words to disappear rapidly before you see the next series of words? That's right here. So initially you're able to see it, because there's a signal. The signal is causing a conformational change in your G protein coupled receptor, causing alpha subunit to be released, and then for GTP to bind. But what's causing the signal to go backwards, in large part, is the intrinsic GTPase activity of T alpha, right? GTP is being hydrolyzed back to GDP. That's right here. In step 8, down here, what do you notice? Look, something got phosphorylated. What was phosphorylated? The G protein coupled receptor. What does that sound like that we just talked about? So the thing that was doing that, the thing that was phosphorylating this was called rhodopsin kinase. What is this analogous to on this slide? The thing that is doing the phosphorylating. That's bark, beta arrestin, beta-adrenergic receptor kinase, right here. So this is a mechanism of desensitization in response to light. You had something phosphorylate your rhodopsin receptor, and then what does it recruit? What does it say to Clathrin? Arrestin. There it is. And what is this going to cause? It's not shown in this figure, but what might you predict? Endocytosis. So there you go. And then here we have, I won't ask you to know it for the exam, but Leah and Ayan, here you are, your retinoid isomerase. Isn't it cool? I mean, I know I'm just talking to myself and I don't know if anyone actually watches these videos, but I just think this is really cool. It is really cool. Well, what about the last figure? What am I trying to show you here? I'm just trying to give you a feel for the different substrates. You have the substrate is light itself, a photon. We've talked about epinephrine in the context of the model GPCR, beta-adrenergic receptor. But there are also all of these random ones that have to do with smell or taste. I honestly don't know what vasopressin does. But you can see what's interesting about this, I guess, is twofold. I mean, it's just remarkable that you have the same archetype of a 7-transmembrane helical receptor that's mediating all these pathways. They all have coupled G proteins, right? And a lot of them even have the same effectors, such as in these 4 cases, they all use adenylyl cyclase to modulate what? To increase the concentration of cyclic AMP. It's interesting. Sometimes you see it. Well, it increases the concentration of cyclic AMP if you have a stimulatory subunit being released. Sometimes it actually decreases the pool of cyclic AMP because you have an inhibitory G protein subunit that inhibits your effector protein, right? And then the last point is sometimes it's not just, when you get later in the pathway, it's not always about phosphorylating or dephosphorylating things through protein kinase A, but there can also be channels that are being impacted that allow important ions such as calcium, sodium, nah, or potassium, okay, from fluxing in and out of the cell. And if you are at all interested in neuroscience, you immediately think, okay, so this must be really important for firing of synapses, the propagation of action potentials. Yep, you'd be right. So on one hand, it's the end game is to cause changes in gene expression, to change the activities of enzymes through phosphorylation that can lead to things like glycogen secretion, or just generally changing the permeabilization, the permeabilized state of the plasma membrane. And it's just amazing that these are all tied together. So this marks the end of the cell signaling section. I encourage you to revisit all of this material on your own. We focused on coagulation and GPCRs. The first question on the in-class activity is asking you to compare and contrast them in 3 different ways. It's listed on Moodle, but really struggle with that and try and force yourself to compare these 2 pathways that we spent so much time going into detail about. And then finish strong. I'm really proud of you guys. And I see that you're trying really hard by keeping up with the in-class activities. And I'm just really wanted to be encouraging to you that you're doing a good job. And this sucks. It just really sucks. Especially, for seniors. It's just life. So thanks for listening. I don't know how long this took, but I did my best to keep it short. Feel free to say hi to me if you would like. If not, I understand. I will see you on Friday when we will talk about the metabolism. We're going to start on metabolism. So I look forward to seeing you then. Stop. End.